Well, thank you, everyone. I will pop a little bit of a slide here. So we're going to be talking about importing and exploring GWAS results with RStudio. I've had great help from Frederick Tan and many other folks to help put this together. And I think we're going to start on the same page in terms of making this a hands-on activity, in the sense that we'll expect that you've all cloned the MaGIC Jamboree 2020 workspace. And this gives you access to links and an important string. So if you want to see the worksheet, you can open that in a new tab. And the worksheet includes information about where the slides are, what a video will be, and takes you through all of this material in a way that can help you catch up if you fall behind, or can just look at later on. The important string to get is this little guy that's going to help us define our new environment. And this script is something you should download. You can click on this and it will download to the ordinary download space on your computer, and we're going to upload it into RStudio in a little while. So let's now capture, actually why don't we do this? We will go to the runtime configuration, pick a custom environment, and yeah, we're going to need to capture this string here, just up to the US, copy that into your clipboard, come back over here to your runtime, pick the custom environment, and pop that string into your container image. We'll take the standard 20 cent per hour compute power situation and hit create for the runtime. Now, I may have messed up a little bit in the sense that you might have to delete an existing runtime to do that. Go ahead and delete that existing runtime and get this one going so that we can run RStudio. It'll take a few minutes to get it going, and while that's happening, we'll look at the slides.
Vince, would you mind sharing how to delete the existing runtime?
Oh, sure.
I don't think we've covered that yet.
Delete the existing runtime. Let's see about that. Let's just get this out of the way here. Come back over here. And to delete a runtime, while it's being created, there's pretty much nothing you can do. We can look at the slides, because the slides actually have full details if that's what you need to do.
Great.
So let us do that. We're assuming you got there. There we are. So I'll pop this up to present mode. Sorry about this. We're at 3 minutes. Okay. So this would be the situation you're probably in before when you're running with a Jupyter Notebook. You've come over to the gear here, and you see this situation, which is your current situation. You hit the delete runtime button, and very rapidly it will query you on whether you should really delete it. You delete it, and now you're in a new situation where you can go back to this gear and say, well, I'm going to pick the custom environment. As I said before, you have a string that you load in for the container image, hit create, and that's the situation we're in now. So we are creating the runtime. If you're not there yet, throw a comment in this chat, and someone who's watching the chat will get back to you on Slack. All right? So I'm going to go back to the subject matter material of our slides, and hopefully we will all be together by the time we get back to that space having to do with runtime. What we want to do is talk about what we want to learn in this session, and that involves interacting with GWAS results. So what we're going to do is use RStudio to produce a Manhattan plot, and we'll make an interactive Manhattan plot that allows us to query the points on the plot for information about what's going on there, and also to slice and zoom in regions of the plot to get a better view of what's going on when it's crowded. Then we'll work with an app to help us search such plots, and also to have hyperlinked tables and graphics in an app. Therefore, we will have explored the data with RStudio, and we'll implicitly show you how to import R notebooks or R Markdown documents and your own data to do this type of work. And the data we're going to be looking at come from this study of glaucoma genetics. This is from the UK Biobank, and this is a 2018 paper in Nature Genetics. And the basic idea that's fundamental is to correctly generate the p-values for association studies so that you can find what parts of the genome help discriminate individuals who have glaucoma from those that do not, and then to try to understand what features of the genome lead to that increased risk. That's GWAS study in a nutshell, getting to a correct representation of this Manhattan plot with corrections for population stratification, et cetera, are assumed to be done. You'll do that elsewhere. You've got some p-values now which are correct, and what we want to demonstrate is how to bring them into RStudio and how to build a Manhattan plot like this, where as I scroll over, this is the static version of it, but as I scroll over points, I can determine what gene is near a given locus that was found to have strong association with the phenotype that we're looking at. This particular Manhattan plot covers chromosome 9, and that's the only chromosome that we'll be looking at in this exercise. Now, those Manhattan plots can be very crowded and unwieldy. You might want to look in the vicinity of a certain gene, and we need help to get there, and that's what the app is for that we will show in a little while. It's easy to produce with a short amount of code, and we can have the same sort of interactive Manhattan plot, but we'll also get a hyperlink table where every locus in here has a row in the table, and we can hyperlink out to dbSNP or to gene cards or to Ensemble or what have you in order to get more information about the things that we're looking at. So these are the tools we'll be trying out in this session. And just to sum up, how we get there is by doing or finding a GWAS. I found the one from UK Biobank. You might do your own. You process and analyze the data. Certainly, you'd want to do that if you've got lots of data. You want to do that in the cloud. And we've shown you tools like Cromwell that can help you take care of that problem. Ultimately, you'll produce a tabular file with one row per SNP, giving the SNP identifier, addresses, p-values, and other sorts of information can be put into that file. And it probably will be produced through a WDL program. We import this association data set into the RStudio session, and then we use R packages, scripts, Markdown, and so forth to do new computations for discovery. So these are the tools we're using: RStudio, Bioconductor, and the Anvil package, which is part of Bioconductor, which will help get the software into your system very quickly and work with the data in the way that I'm going to demonstrate. So that's all I really have to say about the slides to this point. We'll come back to them later on. Let's see where our session is. So this is my little guy here. The runtime is ready. I launch the runtime. This runtime has RStudio baked into it. And when it finally loads up, we'll have the panes of an RStudio session within. This is R version 4. And you see I have a couple of files here which are native to the Terra situation. What we want to do is upload the R Markdown file that we downloaded before. It's called tophitsglauconly.rmd. And we're going to upload that into our RStudio session. Remember, we got that link from the dashboard over here. It's the RMD script. So when you click on that, that goes to your download space. Come back to RStudio, and I have uploaded it now. And you can see that it's in the files collection. If I click on that R Markdown, a new pane is set up which has the code from the R Markdown and the text that describes what's going on. So this is some magical incantations that will describe how an ultimate document could be produced from this R Markdown. And here is a piece of code that we want to run, and we can run it by hitting the green arrow. You can see which pieces of code are running through the dark green part of the run. And we're going through the process of taking this sort of empty R session and populating it with a bunch of packages that will be used in the rest of this set of exercises.
Vince, a small ask from me. Could you hover over buttons before you click them so we can stay up to speed with you?
Sure. So the button there was the little green arrow. So you'll see that again down here. We won't run that one until we're done with this one. So you can see that what we're doing here is we're doing an Anvil install on a bunch of packages that we need, in particular ggplot2, genomic ranges, plotly. These are the tools that are going to help us make the Manhattan plot. And so what's going on now is installation is occurring. You can actually see what's going on if you pop up the console. Each one of these packages is being brought in and installed in my R session. So that's the console, and this is our R Markdown document. And wonderful. We now have all the packages in place that we need in order to do the next things that we want to do. In particular, and this is going to be slightly time consuming, right here we're going to take the CSV file that I described before, which is sitting in a Google bucket, and bring it into our RStudio space and then read it in. So this is chunk number 2, and right now we're copying from this Jamboree bucket into here, and eventually you'll see a new file in my file set, which is the CSV holding all the loci that were recorded in the UK Biobank study of glaucoma. So there it is. You can see it now. We're not going to have to do anything with it because our code will work with that. So we're doing a read.csv of the CSV file. We're now going to filter it because there are some SNPs that they recorded that didn't have an address. And we have no need for those, no use for them. If you need to work with them, you wouldn't do this filtering step. You'd find the ones that are missing addresses and you'd try and find some additional information about them. And so we are now at the point where we have a table, and I'm going to show you what the table looks like. I don't know why, but only if I run this selected line do I see the header of this table, or the head of this table, the first 6 lines, which show me the RS number for the SNP, the allele, the scores. This is exactly what was reported by the UK Biobank folks. Position, strand, p-value. This is what we're going to be working with. Did I hear another question popping up?
Yeah, I was going to pop in real quick. Couple of things. Would it be possible to slow down just a little bit? And
Sure.
would it be possible to jump back to the point where you identified where the R Markdown file was
Absolutely.
and explain how that ended up in your RStudio session?
Sure. I'm going fast because I know I've got to be done at 2:25.
I can go ahead and say that that's not a priority right now.
Okay.
Feel free to take your time.
Okay, well, I'm always trying to meet the prescriptions. So what we have here is a very nice piece of metadata about the workspace. And we can edit this, or people who own it can edit it and see that this is Markdown. Okay? So that is something that the Broad folks have set up, and Fred Tan went and edited it to put in the links that help us get the information that we're interested in. This RMD script is, you can see down the bottom there, is a link to a document which has filetype.rmd, which is in the BioCJam 2020 folder in Google Storage. So when you click on this, this file will go to your downloads spot. It's just doing a default download from whatever browser you're working on. So that's a little bit of local knowledge that you need to have in order to come back to your RStudio session. Is this clear enough? We'll go back to our RStudio session. If you want to upload the RMD, you need to know where to find it. So this is the upload button here. You get to choose a file, and you can go all over your disk. I went to the downloads folder of my own home directory, and see now I have 2 copies of the top hits glauconly.rmd. Okay, so this process is somewhat idiosyncratic. However you want to get your RMD onto your disk and then into your session, that's something that's up to you. But this is the way we did it to try and get everybody on board with a minimum of fuss. So we'll cancel that because I've already got it. Now once you have this here, you can upload it from anywhere. You have this here. When you click on it, it gets loaded into the R Markdown processor that's part of RStudio. I don't even know the name for this pane here, but it basically is something that knows how to process R Markdown and to throw the code into the R interpreter to accomplish what you want to accomplish. In this case, the first piece of code is related to installing software and getting it loaded into your session. We've run it. I'm not going to run that one again. This one is sucking the data out of the CSV file into the RStudio session so that I have a table. And the first 6 lines of that table are here. I will stop because it is a lot, and you know, if this doesn't make sense, we can go back and unpack more of this. But a basic job is to work with this table in order to build a Manhattan plot.
Thanks for backtracking, Vince. It looks like based on the chat that most folks are caught up.
Excellent.
If not, please reach out.
All right. I'm going to do a little more here. I'm going to actually modify this RStudio, this R Markdown document to show you a little bit more. All I want to do is know what the dimensions of this table are, because we haven't discussed that. And it's the dimensions of gstat, which R helpfully tells me about here. And we can run this little new piece of code, and it tells me that I got 1.28 million loci in this file. That's not bad. That's a pretty dense look at chromosome 9. Okay, so that's a nice thing about these R Markdown documents running in RStudio. If you think of something you want to add, you can add a little bit of Markdown and run the line of code that you have added. Just for fun, you can also look at the console, and if you want to run some code that you don't want to put in Markdown, you can do that there. But I won't get into that anymore. Let's just go through one vehicle for interacting with R. All right, so I'm skipping down through the text here, but the text describes all the approaches that can be used to accomplish similar tasks. File on your laptop can be uploaded. You can import it using the files features, or you can have stuff in a Google bucket and use gsutilcp, which is what I used here, in order to get it out of a Google storage bucket. This will be useful if you are throwing things from your WDL job into Google buckets that you know the paths for. Then you can bring that into your RStudio session using code like this. So let's proceed with a Manhattan plot. Right here we are with check, sanity check, simple visualization of a region. So the region I'm going to use is just the first 500 rows of this table. So we'll run this code here. We get green, all is good. For some reason, I have to run that little bit of code again. The second line, I'll run selected lines, and now into my little pane is injected this nice plot. It's a static plot of the first 500 SNPs on chromosome 9, their association with glaucoma. And you can see none of them have really big negative log 10 p-values. Okay, but we're just looking at a small little chunk. So now we want to work with all of chromosome 9. And in order to do that, we're going to do a little bit of magic with bioconductor. We're going to convert the table into a G-ranges. So this is a nice high-level data structure that organizes metadata about all of these SNPs, but is very efficient for working with the actual genomic coordinates. They're on a specific chromosome, they have a specific integer position, and we're going to be able to work with that very nicely in conjunction with gene notation. This is SNP notation. We're going to work with gene notation right here. We're going to run this next little bit of code to get the ensemble version 86. It has all the addresses of gene bodies in build 38. I'll modify that here and run that so you can see what that looks like. Run this little slice of code. And that is a line for every gene in every chromosome. And we're just going to focus on the ones in chromosome 9 when we get down there. Okay? So what we've done here is we've made a table of SNPs. We've plotted it to make sure it makes sense, plotted some lines from it. Then we made it into a G ranges, and finally we got another G ranges which has all the gene body addresses in there, and these are the things we're going to coordinate in order to make our plots in our app. So right now we're going to use a little more code to find overlaps between gene bodies and SNPs that we had at least a 10 to the minus fourth p-value for association. I'm not going to be teaching R here. This is the amount of code it takes in order to find the genes that overlay SNPs that have high association. These aren't going to be the most interesting ones. The most interesting ones will not be in gene bodies. They'll be in regulatory regions. So this is a limitation of this exercise to make it real simple. So we found that there are certain genes here on chromosome 9 that overlay these SNPs. And now we're going to do another full chromosome plot, which is using standard plot in R to show the entire chromosome and the negative log 10 p-values of all the 1.2 million SNPs that we've assembled information on. So that's the quick visualization for a sanity check. And now we're going to do the annotated Manhattan plot. And this is just a boilerplate code that transforms those G ranges into a data frame, which can then be brought into a dynamic Manhattan plot where I can scroll over, hover over the different points, and get the names of nearby genes and the actual RS numbers for these high-scoring SNP. So the code here is very brief. I have to turn the genomic ranges into a data frame so that ggplot can operate on them. The x coordinates are going to be the positions. The y coordinates are the minus log 10 p-values. And then in order to get the hover over text, I have something called the text component of the ggplot aesthetics. And finally, I pump that ggplot through ggplotly in order to get this nice behavior. Not only do I get this hover over, but I can do something like this, which is to slice out a piece of the plot and look at it in a more fine-grained way. And then I can return to the full chromosome by double-clicking. So that kind of functionality I thought was worth showing, a nice feature of RStudio, ggplot, plotly. Comments or questions on that? Okay, we're now going to wrap this up. I'm almost on time. We're going to start this last chunk of code, which is the definition of an app. It's very brief, and there's a lot of stuff wrapped into little components that accomplish what? We're going to run this little guy here, and it pops up a new window. And that window has inputs for gene selection. And one that I like to look at is so this is called a select input. So you can type some clues into what you want to look for. And I just want to look for a gene that I can't remember its name, but I know it has an X in it. Oh yeah, LMX1B. So if I look for LMX1B, I see that there are some SNPs that have 10 to the minus 12 p-value. Pretty amazing. And if I want to look at it in a little more broader context, I can make the radius out to a megabase on each side, and then I can see why people would be excited about this, because it has, a nice concentrated locus with very high association with glaucoma risk. What you can also see here is that LMX1B isn't the only player in the game. There's a pseudogene nearby, the SNP that overlay RP11 here also have very high scores. And then there are other genes that have very high scores in this vicinity as well. So this is the sort of stuff that you have to think about if you're going to try and make a story about why this particular region of the genome shows association with glaucoma risk. These are the sorts of things you may want to work on. And if you want to get some more details, you can click on the table here, which, if I want to focus attention on LMX1B, I can search here, filter the table to those hits that have to do with LMX1B, and then go and find out both about the gene. This is going to link out to gene cards, and about the SNP. So this is a high-scoring SNP. If I wanted to sort, I can find the highest scoring SNP here and then go and see what's going on with that. That takes me out to dbSNP, and I see that it's not reported in ClinVar, it's an intron variant, and that means it's going to be a bit of work to figure out what the functional implications are. So that's it. That is the end of the RStudio session. And I think I can go back to the slides if we are done with this bit of work. All right. So let's take a look through the slides. We have some concluding remarks. We did RStudio. We started it up. We deleted a runtime. We started a new one. Then, using the R Markdown that was put in a Google bucket, we were able to get that top hits Gluck only, run it step by step, and produce the different plots and app that I showed you. Okay, so that's all there for folks to review when necessary. Shutting down the runtime. Maybe we should do that. I think I'll leave it running just in case there's any questions or discussion. But this shows how to shut it down. It's very straightforward. When you deleted, any files that you might have created that you want to keep will be destroyed, so you may want to put them somewhere else, but that probably isn't an issue now. And so to review, we cloned a workspace, brought in a new runtime that includes RStudio, and we brought external files, both a script and a CSV file that we can analyze and guide the analysis with the script. We explored the data using RStudio, tabulated and plotted it using base R, made an interactive visualization using ggplotly, and then ran an app that helps us find more information out based on genes that we're interested in and linked out to other resources, references. So we wanted to take this and suggest some next steps. So for example, if you want to go beyond chromosome 9, it's easy to find CSV files of these summary statistics at this link, the UK Biobank Gene Atlas results. And so you could build a Manhattan plot for a different chromosome. Chromosome 1, for example, has a really strong hit, and it'd be interesting to get that probably. And then to see whether any of these hits have records in ClinVar, which would help you interpret what's going on there. That's one thing to do, and there's a little bit of tricky stuff with respect to taking a CSV file, converting it into G ranges, and allowing us to merge that with gene information. But all the code to do that is in the RMD file. You just have to make certain changes to the file names and what you do with them. Engage with the code to make the app work with any chromosome and ultimately to work with any sort of CSV file that you might want to cook up that has similar types of information in it. And then the question is, can we do a little more biology to help interpret the hits? In particular, the type of thing that you might want to do is a co-localization. So we know we have some big hits, but the question is whether those hits coincide, particularly if they're in regulatory regions, with other SNPs that may regulate, in some sense, expression of genes or other genomic phenomena that, for example, you get out of the expression data in GTEx. So bringing in tools that do co-localization and so forth is definitely on our radar screen. We want to make sure that can be done. And finally, participate in the Slack to propose new features and documents to help advance the platform. So I want to thank Fred and everybody behind the Anvil platform for giving me the resources to help make this. I hope I didn't go too fast, and if there's time to discuss and drill down, I'm happy to do that. Thank you.
Thank you, Vince. In the interest of time, we'll ask folks to provide questions to Vince over our Slack channel. I'd like to mention a few logistics. The materials from today will be made available on the Anvil portal. Once they're available, we will connect with you all and send a reminder and an address to access those materials. I'd encourage you to continue talking to us over the Anvil community Slack, as Vince suggested. This will help us get an idea of what you want to do and how you want to do it, and it'll only help us on the Anvil team progress the platform further. You should have a feedback form in your inbox for the MaGIC Jamboree. Please take a few minutes to fill out this feedback form. We really appreciate it. And finally, we have breakouts starting after closing remarks from NHGRI. And I've just added the link to access those breakout rooms in the chat. They can be found at the top of page 2 on our agenda. Before we hand it over to NHGRI, I'd like to take a minute for us to unmute ourselves and give a hearty round of applause for all of our presenters. They did something very challenging by providing outreach and training over a virtual setting to near 100 people. Please join me in thanking them for their time and effort.
Very well done.